The process of B cell activation is called clonal selection. During this response, a B cell with a specific antigen receptor recognizes an antigen. This cell proliferates to produce a multitude of cells, also called a clone of cells with the same specificity, which differentiate into memory B cells and plasma cells. This process is called clonal expansion. The antibodies produced by the plasma cells bind to antigens to produce the humoral response. In the simplest form of B cell activation, an antigen called a T-independent antigen enters the lymphatic tissue and binds to a B cell receptor, or BCR, on the surface of the B cell. The B cell receptors on a particular B cell are identical and very specific to just one antigen. Once the BCR binds to the T-independent antigen, the B cell becomes activated. It's now ready to proliferate. However, most antigens are not potent enough to activate B cells by themselves. In these cases, the B cell will require a second opinion from a helper T cell in order to become activated. We call these antigens T-dependent antigens. The helper T cell recognizes the antigen as it's presented by the B cell and secretes cytokines to tell the B cell to become activated. Once a B cell becomes activated, it proliferates or divides, forming a clone of cells specific to the same antigen. While some of these B cells differentiate into long-lived memory B cells, most differentiate into plasma cells, which secrete antibodies against the antigen that activated them. The antibodies can enter the bloodstream and travel throughout the body. Plasma cells only live for a few days, but each one can produce about 2,000 antibodies every second, resulting in enormous quantities of circulating antibodies.